Turns out, Chemo-Brain and the amino acid that might stop it thought cancer patients beat the disease. Then their brain stops working. They forget why they walked into a room. They can't follow a conversation. They call it Chemo-Brain, and for decades we thought it was just the price of survival. But what if it wasn't? What if a single amino acid, something your body makes naturally, could stop it before it starts? Here's the question. If we could prevent cognitive decline in cancer survivors with something as simple as glycine, why aren't we doing it yet? Chemotherapy is brutal. That's the point. These drugs are designed to kill cells, and they're devastatingly good at their job. The problem is, they don't discriminate perfectly. Cancer cells die, but so do some of the healthy cells around them. For decades, oncologists accepted this as unavoidable collateral damage. You trade your hair, your energy, sometimes your organs, and you get your life back. That was the deal. Except it wasn't always true. Survivors kept reporting something strange. Their body was healed, but their mind felt broken. Memory gaps, brain fog, difficulty concentrating. The medical establishment largely shrugged. Chemo-Brain was real, but vague. It was attributed to trauma, stress, anesthesia, everything except the obvious culprit. Scientists suspected oxidative stress and inflammation in the brain were responsible, but proving it and fixing it were two entirely different problems. This is where a team of researchers from Pakistan decided to test something radical. Led by researchers at Ayub Medical College and Khyber Medical University in Pakistan, a multi-institutional team published their findings in June 2026 in the Annals of Medicine and Surgery. Their names are worth knowing: Haris Mahmood Ompir Kazmi, Nasir Ullah, Mohammed Ikram, and the rest of the collaborative team. These weren't researchers in a wealthy lab with unlimited funding. They were working in Pakistan, where resources are tighter, collaboration is crucial, and the questions they ask have to matter. This study mattered because it asked something nobody had seriously tested in mice before. Could glycine, a simple amino acid, actually protect the brain from chemotherapy toxicity? The setup was straightforward but rigorous. 25 healthy adult male mice, divided into 5 groups, 28 days. Some mice received cisplatin, one of the most common and most toxic chemotherapy drugs, injected into their abdomens every 4 days. Some received glycine every single day through a subcutaneous injection. Some received both. Some received neither. Some received glycine first, then cisplatin, to test whether protection had to be simultaneous or could kick in later. Then the researchers tested their brains. Think of it like this. You're testing whether a security system can protect a building from a wrecking ball. The wrecking ball is cisplatin. The security system is glycine. You can install it before the ball swings, or after, or not at all. Then you measure how much damage the building takes. The wrecking ball worked. Cisplatin alone devastated the mice's cognitive function. On the Y-maze test, a simple measure of working memory where mice navigate a maze and hopefully don't repeat the same wrong turn twice, cisplatin-treated mice performed catastrophically worse. The difference wasn't subtle. It was statistically significant at p less than 0.001. That's not just real. That's unmistakably real. The Morris Water Maze test measured something harder. Actual learning. Mice were placed in water and had to find a hidden platform. Normal mice get faster at finding it. Cisplatin-treated mice didn't. They fumbled around, taking longer to find safety each time. Again, p less than 0.001. The drug worked exactly as researchers expected. It poisoned the brain's ability to remember and learn. Then the glycine kicked in. Mice that received glycine at the same time as cisplatin, that's the crucial part, showed the greatest protection. Their Y-maze performance recovered significantly. Their Morris-Water maze latencies improved. They weren't totally spared. Cisplatin is nasty, but the damage was substantially reduced. The concurrent treatment was the most effective intervention the team tested. But here's the part that matters for real humans. Delayed treatment with glycine also worked. When researchers gave cisplatin first, then started glycine afterward, cognitive decline was partially reversed. Not completely, but meaningfully. This suggests that glycine might work not just as a preventative, but as a rescue strategy, a way to repair damage that's already started. The mechanism is probably what researchers expected. Glycine is an inhibitory neurotransmitter. It calms things down. It reduces oxidative stress and inflammation in the brain. Cisplatin cranks both of those up to 11. Glycine turns the volume back down. Simple. Elegant. And it actually works. For cancer patients, this changes the conversation. Chemo-Brain has been treated as an acceptable side effect because we had no alternative. We had no idea how to prevent it. Now, in mice at least, we have proof that a cheap, naturally occurring amino acid might prevent it entirely if given alongside treatment. Glycine is already safe in humans. It's used in medical settings, and your body produces it constantly. The doses in this study were substantial. 1 gram per kilogram of body weight daily, scaled to humans, would be roughly 50 to 100 grams per day. That's higher than normal, but not unprecedented in clinical settings. The honest limitations. This is mice. Mice are not human brains. Cisplatin affects mice differently than it affects us. The timeline was only 28 days. Real patients get treated for months or years. We don't know if glycine's protection would hold up under those conditions, or whether it would interfere with the cancer-killing effectiveness of the drug itself. That last one is crucial. If glycine weakens chemotherapy's ability to kill cancer cells, the whole strategy collapses. The study doesn't address that. But the bigger picture is this. For the first time, we have experimental proof that you might not have to choose between destroying cancer and preserving your mind. This finding sits in a growing body of research asking a question that's been criminally overlooked for too long. What if we could make chemotherapy safer without making it weaker? For decades, toxicity was just accepted. Side effects were just the price. But this study, and others like it, suggest that might be lazy thinking. There might be interventions hiding in plain sight. The real question isn't whether glycine works in mice. It probably does. The question is why, if we suspected this for years, did it take until 2026 for someone to actually test it? And more urgently, how many other simple interventions are we not testing because we've accepted that cancer treatment has to destroy your quality of life? That one molecule might prevent thousands of cancer survivors from years of cognitive suffering. That's extraordinary. So why isn't every hospital testing this in humans right now? Turns out, scientists have been busy. And they're not done yet.